Alright, so let's talk about the prokaryotic cells in this particular video. So let's start from the very basics. Like in this video, we'll be dealing with the basics of the prokaryotic cells and then we'll move on to the complexity. And here I have taken very simple diagram of the prokaryotic cell, which is the bacterium. And when I say, okay, just forget everything. When I say prokaryotic cells, what do you understand by the term prokaryotic? Let's break the word for your ease, because biology is a vast subject. So in this subject you deal with various types of words which can be breaked easily. Okay, like for example, cytology. Cyto means cell and logos means study. So cytology means the study of cell. Similarly, here you have the word which is prokaryotic. Let's break the word. So this word can be broken into two parts, that is pro and karyon. Okay, pro and karyon. Okay, now looking at the literal meaning of these words, so pro means early, primitive, before. Okay, and karyon means nucleus. Now you might be getting confused, like when I say primitive nucleus, so what is the meaning of this word term primitive nucleus or before nucleus, something like that? So primitive nucleus, before nucleus, early nucleus. So here when I've shown you the diagram of bacterium, which is a prokaryotic cell, here you can see that the nucleus, you can see the chromosomes here, right? Here you can see that there is no nucleus found. Okay, there is no nucleus found. That the chromosomes are lying freely in the cytoplasm. So when I say primitive nucleus, means the material which is present inside the nucleus that is lying freely in the cytoplasm. This is the literal meaning, or the simple meaning of prokaryotes. Okay, so bacterium, blue-green algae, or the cyanobacterium, these are the examples of prokaryotic cells. Okay, now that you people know like what is prokaryotic, let's deal with the structure of the prokaryotic cells. Here I have taken the example of a bacterium. This is a very simple diagram of a bacterium. Dealing with the structure, let's move from outside to inside. Okay, so here you can see that bacterium is surrounded with some yellow, just talking about this light yellow color layer. So this is surrounded with some membrane or layer, right, which is of light yellow color. So here they have given the name as cell capsule. So now what is this? Okay, so this is known as the glycolite in biological language, more diverse language if I talk about. So it is known as the glycolite. Okay, and glycolites are also of two types. One, we have the thicker one, which is known as the capsule, and then we have the thinner one. Which is a kind of loose sheet, which is known as the slime layer. Okay, but right now you can just focus on that bacterium is surrounded with some kind of a capsule. Okay, like do you know what Roman dolls are? We have one, under that we have one, inside that we have one, inside that we have one. So this is kind of similar structure. Okay, familiar structure. Here when you see that the outer layer is a kind of capsule, inner to that lies some dark yellow kind of layer, that is the cell wall, the murine cell wall. Okay, which is composed of a material which is known as peptidoglycan. Murine, we can also say murine. So which it is composed of a material called peptidoglycan. Let me write it for you. Peptidoglycan. Okay, it is composed of some material called peptidoglycan, also known as murine, which is here mentioned, murine cell wall. Okay, now what is this? This peptidoglycan are actually the macromolecules. Okay, they are the macromolecules which are composed of amino acids and the amino sugars. Okay, now these amino sugars are also of two types mentioned here, NAG and NAM, and we have the acetyl glucosamine, and we have the acetyl muramic acid. Okay, now that you know what the cell wall is here. Now you know this moraine cell wall, the cell capsule. Inner to that, this light blue colored fluid kind of structure which is present, that is the cytoplasm. It is the place where all the organelles and everything lies. Okay, so here, and there's a speciality of the prokaryotic cells, the bacterium especially, like the organelles which are present here, they are not membrane bound, they are lying freely in the cytoplasm. Okay, they are lying freely in the cytoplasm. Okay, now you know what cytoplasm is. Now this we are talking about, this blue kind of complex structure. These are actually the this is what nucleoid is. Okay, the the chromosomes which are present, they are not membrane bound. They are not having the nuclear membrane like in a normal cell. When you see that this is a cell wall, then inner to that we have the cytoplasm, the cell membrane and the cytoplasm, and then inside to that we have a nucleus, and the DNA, the genes, the chromosomes, all is present inside this nucleus. But when you look at the prokaryotic cells, here you find that there is no nuclear membrane present, and the chromosomes are lying freely inside the cytoplasm. They are not membrane bound. Everything is so hodgepodge kind of in the prokaryotic cells. Okay, so this is what you can see here. Getting my point? Okay, now this red kind of complex little structures that you find is plasmid. We'll discuss about this as well. Now the last structure which is present here is the flagellum. Flagellum is a kind of protrusion which is present, which is flagellum is kind of protrusion which is coming out of the bacterium, the body of the bacterium. It helps it to move, and we'll also discuss about flagellum in detail, like the basal body, the hook, the filament and everything. So stay tuned till the very end. Okay, now focus on this picture. And I was talking about the flagellum, right? Now here you can see how a flagellum looks like. This is also a very simple diagram of bacterium. Here you can see that this is the flagellum, most probably. If I try to zoom it a bit, here you can see that this area from which it is being attached, that is known as the basal body. Now this red part is known as the hook. This red part is known as the hook, okay? And this blue kind of structure, this structure is known as the filament, and all together this entire structure which is coming as a protrusion, coming out like a protrusion from the bacterium's body, that is known as the flagellum. So this is what it is like. Okay, yep. Now when we see it more clearly, so the cell wall, we were talking about the cell wall, right? So peptidoglycan, it is composed of peptidoglycan, which is moraine, and I told you about the macro molecule composed of amino sugars and amino acids. And the amino sugars, they are the N-acetyl glucosamine, NAG, and N-acetyl muramic acid, that is NAM. Okay, now when you see a closer or zoomed view of this, so you find a cross-linked kind of structure which gives strength to the cell wall, the cross-linked peptides, they give strength to the cell wall. And the function, now dealing with the function, why this peptidoglycan is present. So the means why cell wall is composed of this peptidoglycan or moraine. So the function is that it provides protection. Okay, it provides protection. It gives definite shape to the bacterium, because cell wall, the function of cell wall is to give shape, right? That stiffness or that shape to the cell. So this is the function of this peptidoglycan as well. All right. Okay. Okay, and there is one more thing that I wanted to mention, is that we were talking about the flagellum, right? I'm just writing the short form. And we learned that it has three parts, which is the filament, hook and the basal body. So now there is one more factor, that sometimes in the bacteria you find very little kinds of protrusions coming out of the bacteria. They are not the flagellum. First of all, let me tell you, they are not the flagellum, because they are kind of very small structures, very minute structures which are coming out of the bacterial body. So they are known as the pili, most probably the pili or the fimbril. Okay. Now this paleofibril is kind of attachment. Okay, it is present on the outer surface of the bacteria. It is present on the outer surface of the bacteria and helps it to stick on the surface of anything. Okay, it helps it to stick onto the surface of anything. And we also learned about the capsule which is surrounding the bacterial body, right, which is known as the glycolite. So glycolite also has the property which helps the bacterium to stick onto the other body. Getting my point? Okay, now dealing as we were talking about the flagellum, right, based on the bacterial classification is also based on the number of flagellum present. Okay, now we will talk about the number of flagellums present as well. Okay, all right then. Okay, so we have the bacterial classification also based on the types of flagellar arrangement. Like you can see in this picture, the first one, here we have no flagella present. So that is why it is known as atricus. Atricus, okay? And then in the second one, you can see that there is only one flagella arising from the body. That is why it is known as monotrichous. Mono means one, okay? Then in the second one, we have the flagellum arising one from each side, from both the sides. Okay, so that is why it is known as amphitrichous. Amphi means two. Okay, so like we have the amphibians, the animals which can live on both land and water. So that is why here amphi means two and trichous is the flagellum. So and amphitrichous means the flagellum arising from both the sides of the bacterial body. Then in this picture, it's kind of bit complex. It resembles the pili and the fibril we talked about, but it is not pili and fibril. It is also kind of flagellar arrangement, which is known as the peritrichous, and it is there on the entire body of the bacteria. Then in this we have the flagellum which are arising from the same point of the body. So that is why it is known as the lophotrichous. Okay, it is known as the lophotrichous. Then we have this, in which we have we see that the flagellum are arising from both the sides of the body, and this is known as cephalotrichous. Okay, cephalotrichous. So these are the 6 classification of bacterial arrangements based on the flagellar arrangement. Bacterial classification based on the flagellar arrangement. You can just take a clearer view. This is what it looked like. Now look at this structure. This structure is kind of a bit complex, a structure of a bacterium, a bit complex only. Now talking about the cell organelles of the prokaryotic cells, so there are no membrane bound organelles present, because as I told you that in a bacterium, or you can say any prokaryotic cell, whether you talk about the cyanobacteria, so most probably they have no membrane bound organelles present. The internal structure is very hosh-posh kind of. Okay, there are, like the nucleoid also you can see, the chromosomes are not bounded inside the cell nuclear membrane. They are freely lying inside the cytoplasm. Okay. Now when I talk about mesosome, which is a special structure present, like this is a clearer view of a mesosome. Here you can see that mesosomes are nothing but extensions of the cell membrane or the plasma membrane. Like this is the cell membrane or the plasma membrane, and they are just the extensions of the cell membrane or the plasma membrane. Now what is their function? So their function is the synthesis of the cell wall. Okay, they increase the surface area, the DNA replication. This is a very, very important point. DNA replication and distribution to the daughter cells, respiration, secretion. Okay. All right. Apart from all that, we also have the inclusion bodies present in the inclusion bodies present in the bacterium. Okay, so they are freely present in the cytoplasm, as all the cell organelles are not membrane bound and they are freely lying inside the cytoplasm. So they may contain the phosphate granules, the glycogen granules, the fission granule, and etc. They also might contain the gas vacuoles and all. Okay, apart from all this, now we have a special character which is known as the ribosome. Ribosome is also a cell organelle. We are talking about the ribosomes now. We are dealing with the ribosomes now. Okay, ribosomes. They are kind of special cell organelles, so it's not membrane bound, but there is exception that it is also present in the eukaryotic cells. We'll discuss about eukaryotic cells very soon, inshallah. But here we are talking about the prokaryotic cells, but this is an exception because in eukaryotic cell everything is so well defined and well structured. Okay, like this I'll give you an example. This prokaryotic cell is just your Nokia phone, and your eukaryotic cell is a developed version. It's your iPhone. Getting my point? Okay. Now talking about the ribosomes, it's not a membrane bound organelle, but it is present in the eukaryotic cells as well and in the prokaryotic cells as well. But here we are talking about the prokaryotic cells. So now what are these ribosomes? They are the cell organelles and also the they are the cell organelles and also the site of protein synthesis. Okay, they are the cell organelles and also the site of protein synthesis. Protein synthesis means protein is being synthesized here, is being prepared or made here. Getting my point? Okay, now to understand this in a better way, let's take an example. Now consider this is your consider this to be your chromosome. Okay, chromosome. Now here the genes are present, right? Here the genes are present, and genes are containing the messages. All the messages and everything is contained inside the genes, right? Now these chromosomes, they are having the genes, and genes are having the important message. Now this message will be transformed, it will be transferred through the mRNA to the protein characters, to the ribosomes. Okay, it will be transferred to the ribosomes through the mRNA, messenger RNA. Okay, I'm repeating it again. From the gene or the chromosomes where genes are present and genes are containing the messages, the message will be transferred to the ribosomes where the protein is synthesized through the mRNA, that is the messenger RNA. So here the protein characters are present. Now these ribosomes are moving like this. Ribosomes are here on this is the mRNA, and the ribosomes are present like this. Okay, they are present like this. Getting my point? Okay. Now these their function is that now when the message will reach there, I know that you people are getting confused. Okay, let's take a very simple example. Now if there is a notice with any teacher of your school, okay, kind of your biology teacher. Now your biology teacher is personally told that tomorrow in the class there will be only 4 periods held at the place of 9 periods. Okay. Now only the biology teacher knows that tomorrow 4 periods are going to be held, but that message is not transferred to the other students or the teachers or anybody. Nobody knows except the biology teacher that 4 periods are going to be held today. So will anybody know, will tomorrow 4 periods be held or 9 periods will be held as usual? Obviously 9 periods, right? Because nobody is aware that there are some changes in the curriculum or the timetable. Okay, now similarly here this gene is the school management and all. Okay, here message or the decisions are being taken place, decisions are taking place, and through the mRNA or through some messenger or something, it is transferred to the ribosome. Now the ribosome is going to type the message or prepare the notice, and this notice is going to be circulated to the students and all the teachers and all. So this ribosome is the typewriter who is writing the notice. So this is synthesizing the message. It is synthesizing protein. Okay, it is synthesizing protein, and this protein is being transferred to in the form of a notice, most probably in the form of a notice to everybody, and now everybody is aware that tomorrow 4 periods are going to be held at the place of 9 periods. Easy? Yep. Okay. More point that I would like to mention is that, I was talking about the RNA, and I told you that the arrangement of ribosomes is in this kind of way on the RNA, right? So if multiple ribosomes are there on an RNA, so this multiple chain of ribosomes on a mRNA is known as polyribosome or polysome. It is known as polyribosome, ribosome, ribosome or polysome. Okay, so this is what it is like. And there is one more factor, like in a structure of a ribosome, if you look at the structure of a ribosome, so you see that the upper portion is kind of smaller as compared to the lower portion. You'll study about it, is of 70s, you'll study about this later on, inshallah. And this is what a prokaryotic cell is like. And on, you know what, I was talking about the plasmids also. There was a factor which I mentioned in the between of the video, which was the plasmids. Like in the bacteria we have kind of small structures which are called the plasmids. So they are very significant in the biotechnology. They're kind of tool in the biotechnology, and they're actually the extra chromosomal DNA, the plasmids most probably. Okay, and one more thing that I missed to mention in the previously was that when we were talking about the chromosome, so this folding kind of arrangement of a chromosome, this is actually known as the scaffold. SCAFFOLD. Okay, so this is known as a scaffold. Okay, so this is it. In this particular video, we talked in detail about the prokaryotic cells. So the topic is completed, alhamdulillah. I hope you enjoyed the video. Jazakallah khair for watching. Allah Hafiz.